The process of sperm production in the male body is a remarkable journey, taking place deep within the testes, specifically in tightly coiled structures called seminiferous tubules. To fully appreciate the complexity and precision of this biological marvel, let's embark on a step-by-step exploration of how sperm are produced, matured, and made ready for their vital role in reproduction. Imagine this journey unfolding in three-dimensional space as we shrink down to microscopic size and travel alongside the developing sperm cells. Our voyage begins in the testicles, the oval-shaped organs located in the scrotum. Within these organs, over 500 meters of seminiferous tubules are packed into each testicle. These tubules are the factories of sperm production, a process known as spermatogenesis. The walls of these tubules are lined with specialized cells called spermatogonia, which are the sperm stem cells. These cells are the progenitors, the starting point of sperm development. At the onset of puberty, usually between the ages of 12 and 16, hormonal signals from the brain, the hypothalamus, and the pituitary gland activate the testes. The pituitary gland releases two critical hormones: luteinizing hormone, LH, and follicle-stimulating hormone, FSH. LH prompts the Leydig cells in the testes to produce testosterone, the main male sex hormone, while FSH stimulates the spermatogonia to begin their transformation. Imagine drifting along the inner wall of a seminiferous tubule. Here, you witness the first stage, spermatogonial mitosis. The spermatogonia, positioned near the outer edge of the tubule, divide by mitosis, a process that results in two identical daughter cells. One remains as a spermatogonium, ensuring a steady supply of stem cells. The other advances to the next phase, becoming a primary spermatocyte. This journey from spermatogonium to mature sperm is not instantaneous. It takes approximately 64 to 74 days from start to finish. The timeline is meticulously orchestrated, with each stage building on the last. As the primary spermatocyte moves inward toward the center of the tubule, it undergoes meiosis, a special type of cell division that reduces the chromosome number by half, ensuring that sperm carry just one set of genetic instructions to combine with the egg set during fertilization. During meiosis I, the primary spermatocyte divides to form two secondary spermatocytes, each with half the usual number of chromosomes. These secondary spermatocytes rapidly enter meiosis II, dividing again to form four round cells called spermatids. At this stage, the cells are still unrecognizable as sperm, lacking the distinctive tail and streamlined shape. Now, in the heart of the seminiferous tubule, a process called Spermatogenesis begins. The round spermatids undergo dramatic physical changes, transforming into sleek, elongated sperm. Their DNA condenses tightly within the head, while a cap-like structure, the acrosome, forms to help penetrate the egg during fertilization. Simultaneously, the cell grows a whip-like tail or flagellum, essential for swimming. Excess cytoplasm is shed, and the midpiece fills with mitochondria, providing the energy needed for the long journey ahead. Throughout this transformation, Sertoli cells, large, nurturing cells lining the tubules, play a crucial role. They provide structural support, nutrients, and help remove the waste created during the remodeling of the spermatids. Sertoli cells also create a protective blood testis barrier, shielding developing sperm from harmful substances and immune cells. Once the transformation is complete, the immature sperm are released from the Sertoli cells into the lumen, or central channel, of the seminiferous tubule. However, they are not yet fully functional. The sperm are now called spermatozoa, but they lack motility and the ability to fertilize an egg. From the seminiferous tubules, the sperm are gently propelled by fluid and contractions into a network of tiny ducts called the rete testis, and then into the epididymis, a long coiled tube sitting atop each testicle. The epididymis is divided into three parts: the head, body, and tail. Here, sperm undergo a final maturation process that takes about 2 weeks. As we travel with the sperm through the epididymis, we see them gradually gain motility and the capacity to fertilize an egg. They interact with proteins and enzymes secreted by the lining of the epididymis, which further modify their membranes and prepare them for their ultimate purpose. By the time they reach the tail of the epididymis, they are fully mature and ready for ejaculation. Altogether, from the first division of a spermatogonium to a fully functional spermatozoon, the process takes about 2.5 to 3 months. The testes are continuously producing sperm, with millions being generated every day to ensure a constant supply. When ejaculation occurs, mature sperm are mixed with fluids from the seminal vesicles, prostate gland, and bulbourethral glands to form semen. This fluid nourishes and protects the sperm as they travel through the male reproductive tract and into the female reproductive system during intercourse. The entire system is delicately balanced by hormones. Testosterone, produced by the Leydig cells, maintains the process of spermatogenesis and sustains male secondary sexual characteristics. FSH and LH, continuously released by the pituitary gland, keep the whole operation running smoothly. If any part of this hormonal axis is disrupted, sperm production can slow or stop, leading to fertility issues. Reflecting on this timeline, we see an intricate dance of biology: stem cells dividing and differentiating, specialized support cells nurturing the process, and hormones orchestrating each step with precision. The male body's ability to produce billions of sperm over a lifetime is a testament to evolutionary ingenuity and the importance of reproduction. The journey of sperm production is not only a story of cellular transformation, but also one of resilience and renewal. Every day, new generations of sperm begin their journey, ensuring that the potential for life continues, unbroken, from one generation to the next. Through this remarkable process, the male body fulfills its vital role in the perpetuation of our species.